Okay, let's get started. Hello, my name is Alan Hudson from the Immuno-Oncology Translational Network. I also serve as chair of biostatistics and bioinformatics at Roswell Park Cancer Center. I'd like to make a few points about today's seminar. In supporting the next generation of cancer researchers, today's Cancer Moonshot Seminar will feature researchers from talented junior investigators, otherwise known as early career scientists. These are abstract-driven talks nominated by Cancer Moonshot investigators and selected by NCI staff. I also have a few bookkeeping items to go over. Questions will be responded to at the end of the second presentation. Please enter your questions on the Zoom Q&A box. The audience can prioritize the questions by clicking on the thumbs up button. Closed captioning is available. The closed captioning for today's webinar can be accessed by clicking on the live transcript option in the Zoom menu at the bottom of your screen. Instructions will also be provided in the chat box. The recording of today's webinar will be available a few weeks after today's date. With that, I'd like to turn the proceedings over to our moderator, Dr. Hernandez-Lopez. Dr. Hernandez-Lopez is an assistant professor in the departments of bioengineering and of genetics at Stanford University, a member of Stanford BioX, and the Chan Zuckerberg BioHub investigator. Dr. Hernandez-Lopez received his Ph.D. in chemical physics at Harvard University. During his postdoctoral research at UCSF, Dr. Hernandez-Lopez was a fellow of the Cancer Research Institute and UC Mexis. I hope that's how you do the acronym there. At Stanford, his work focuses on engineering novel T-cell therapies for cancer treatment. Dr. Hernandez-Lopez is also co-founder of Clubes de Ciencia Mexico and Science Clubs International, a nonprofit organization that aims to expand access to high-quality STEM education and collaborative international networks. Dr. Hernandez-Lopez.
Thank you, Alan. Welcome, everybody. I'm very excited to be here, and it's my pleasure to introduce the first speaker of today's series, Dr. Robert McGray, Assistant Professor of Oncology and Immunotherapy at the Roswell Park Cancer Center. Dr. McGray obtained his PhD at McMaster University working on therapeutic responses to cancer vaccines. Then he did a postdoctoral fellowship at Roswell Park, where he worked on combining oncolytic biotherapy with cancer vaccines and adoptive T-cell transfer. Then he went to work in a clinical stage company developing cancer immunotherapies and came back to Roswell Park in 2020. His lab is focused on developing immune cell engagers to target tumor cells and cells within the tumor microenvironment. Looking forward to your talk, Dr. McGray.
Okay, hello, can you hear me?
Yep, we're good, all good. You need to put in presentation mode though, Robert.
Okay, see the slides as well?
Yes, now you're good.
Perfect, okay, great. Well, thank you very much for the introduction and thank you very much for the opportunity to speak here today. So today I'm going to tell you about work that's being done in our group here at Roswell Park, working on engineering T cells to secrete bispecific T cell engagers or BiTEs as a form of adoptive T cell therapy for ovarian cancer. This is my relevant disclosures for the talk today, just related to a patent that I'm a co-inventor on related to the T-cell technologies I'm going to discuss. When we think about treating ovarian cancer with immunotherapy, I think context is important, and that is the majority of ovarian cancer patients are diagnosed with advanced late-stage disease, and following frontline surgery and chemotherapy, most of these patients are going to eventually develop relapsed and recurrent disease. And so really these are the patients that we think about that are going to probably be receiving immunotherapy as subsequent lines of treatment. It's long been appreciated that intraepithelial CD8 T cells in ovarian cancer are prognostically favorable. And this is data generated here at Roswell Park almost 20 years ago from Dr. Kunle-Ondadzi's lab, demonstrating this phenomenon. But somewhat paradoxically, well, immunotherapies are quite impressive in terms of response rates and other indications. Really, in ovarian cancer, the response rates are very modest for immunotherapy. So despite this high level of T-cell infiltration in some patients, these patients don't go on to respond to immunotherapies. And more recent data suggests that this could be because the majority of these T cells that infiltrate tumors actually lack tumor specificity and are not reactive against the tumor, and instead are comprised predominantly of bystander T cells. And so the question that we've been working on is whether we can effectively redirect these bystander TILs and repurpose them for effective tumor attack. The approach we've been investigating is use of bispecific T-cell engagers, or as they're commonly referred to collectively, BiTEs, which are synthetic proteins that are engineered to recognize an antigen of interest and then also the CD3 complex. And in this way, you can bypass the normal TCR, MHC, or HLA interaction that's required to activate T cells. So typically, BiTEs are generated by linking the antigen binding domains from two antibodies, typically in the form of scFvs, via short flexible linkers. And in this way, you can create an artificial immunological synapse between a T cell and a target cell of interest. In this way, you can redirect the T cell regardless of specificity in order to generate a polyclonal response from the endogenous pool of T cells. And similar to other approaches that use CARs, there's no MHC or HLA restriction for this approach. There are of course, some limitations related to the use of BiTEs. First, you really require T cell infiltration given the mechanism of action for this therapy. So if the tumor lacks T cell infiltration or sufficient T cell infiltration, then the attack on the tumor is not going to be robust. Secondly, in their simplest form, BiTEs typically lack an Fc domain, which essentially means they're going to not persist for very long post-infusion, typically in the order of in the order of hours versus days in terms of half-life in circulation, which means essentially you either need repeat or continuous infusions to reach therapeutically meaningful levels of exposure. And of course, BiTEs also need to co-localize with T cells to drive an anti-tumor immune response. To get around some of these challenges, we've been engineering T cells to secrete BiTEs in our group. We refer to these collectively as BiTE T cells. And BiTE T cells can function in a couple of different ways. When the T cell secretes a BiTE, it can either bind back directly to the T cell that is secreting the BiTE and redirect that T cell for tumor attack, but it can also redirect and engage bystander T cells to drive productive anti-tumor immunity. So in this way, leveraging the power of these bystander T cells. And so our feeling is that this can overcome multiple challenges associated with BiTEs. First, because the adoptively transferred T cells are going to expand following infusion, this acts as a durable source of BiTE secretion, removing the need for these repeat or continuous infusions. Secondly, you automatically generate an increased frequency of T cells that co-localize with BiTEs because the T cell itself is the factory for BiTE production. And third, as already noted, you can engage endogenous immunity directly, which essentially offers a unique mechanism of action compared to other forms of ACT that would use either TCRs or CARs as the base form of therapeutic. In ovarian cancer, we focus on targeting folate receptor alpha, and the T cells I'm going to discuss are called FRB or FRBH T cells. Folate receptor alpha is overexpressed in the majority of ovarian cancer patients. This is data here from TCGA, illustrating that ovarian cancer has the highest level of folate receptor alpha among all indications shown. Furthermore, folate receptor alpha expression is typically increased with disease relapse and is associated with chemotherapy resistance. So if we think of the patients that are likely to be treated with immunotherapy, it puts us right in the sweet spot of those patients. Additionally, because there's a long history of targeting folate receptor alpha with a variety of different therapeutics, there's a long track record of showing that it's both immunogenic but also well tolerated clinically when targeted in patients. So I just want to show here that we can generate FRBH T cells with high efficiency using retroviral transduction. And here we're using a GFP reporter that's included within the retrovirus to track transduction efficiency. When we co-culture FRBH T cells with folate receptor alpha expressing target cells, here we're showing SCOP6, which is a cervical cancer cell line. We have potent target cell killing even at very low E/T ratios. And furthermore, when we test SCOP6 xenograft models and we infuse FRBH or control engager armed T cells, we see very rapid and deep regressions with the FRBH T cell therapy. So based on this data, we then developed a translational model for testing FRBH T cells in the context of true ovarian cancer patient tumors. And so to do this, we accessed samples from the Roswell Park GYN tissue bank. These were stage 3 or 4 serous ovarian cancer patients. And here we're using their ascites samples, which contain a mix of immune cells and tumor cells. We then co-cultured these samples with either FRBH or control engager armed T cells. And then at 48 hours, we looked at various parameters of functionality or efficacy, including target cell killing, engagement of the armed and engineered T cells, and also whether or not we were engaging the endogenous tumor-associated lymphocytes or TALs that were part of the patient's own immune system. When we look first at the level of expression for folate receptor alpha across these patients, we found that really patients could be placed into 3 different categories, either low expression for the target antigen, intermediate, or high. And we found that 7 out of 10 patients had greater than 20% folate receptor alpha positivity. When we then look at response by these different levels of antigen expression, we see that the high patients respond very robustly. And so here what I'm showing is overlaid data. So the bars are the tumor cell counts following co-culture, the red being the antigen positive cells, black being antigen negative tumor cells. And then the line overlaid is the amount of interferon gamma produced within the co-culture during the assay period. And so for the high patients, we get very robust clearance of folate receptor alpha positive tumor cells, and we get corresponding increases in interferon gamma production following addition of FRBH T cells. We see very similar data for folate receptor alpha intermediate patients, although the magnitude is slightly less. And for the low patients, we essentially see very low reactivity, in some cases with control engager armed T cells and FRBH T cells looking very similar in these assays. And so what this told us was some information around the types of patients that might benefit from treatment with FRBH T cells. As part of these studies, we also want to mechanistically understand which T cells were actually driving the response. And so in these co-cultures, we pre-labeled the engineered T cells with cell trace violet, which then allowed us to separately interrogate the endogenous T cells, which are cell trace violet negative, the engineered T cells, which are GFP positive and cell trace positive, and then the bystander T cells, which are cell trace positive but negative for GFP. As we expected from our preliminary testing, we saw very robust activation of the FRBH T cells in response to co-culture with folate receptor alpha expressing tumor cancer patient samples. And we saw essentially no activity from the control engager T cells. Additionally, what we observed was that the endogenous T cells are also activated through BITE secretion. And so here with the FRBH T cells, we see a very clear activation of endogenous T cells in comparison to what we see compared to the control engager armed T cells. And so this let us know that the patient's own T cells were also contributing to the therapeutic response. To better characterize the nature of this response, we moved to an immunocompetent mouse model, which would allow us to really understand the role of host immunity in its interaction with the engineered and adoptively transferred T cells. And so here, we're using an ID8-derived model. This is an aggressive variant of ID8 that's also been engineered to express human folate receptor alpha. And here we're using a chimeric T cell engager that binds human folate receptor alpha and mouse C3 epsilon. And so here, as you can see, that when we implant tumors intraperitoneally, give local regional delivery of T cells, the FRB T cells can control the tumors and lead to improved survival compared to the unarmed control T cells. We've also found that how we precondition these T cells is important. So if we use IL-2 and IL-15 to produce a less differentiated T cell phenotype, we can see improved efficacy compared to the use of IL-2 and IL-7, which produces a more effector-like T cell population. When we looked at animals to understand the nature of this response and the improved outcome with the 215 stimulated T cells, what we observed was that when we look at what cells actually infiltrate into the solid tumor, so here showing TILs, we saw no difference between the 2.7 and 2.15 stimulation. Where we did see the difference was in the peritoneal cavity, here denoted as peritoneal wash, and these are the tumor-associated lymphocytes, where here we see a much improved persistence of the FRB 2.15 T cells compared to the 2.7 T cells. Furthermore, these FRB 2.15 CD8 T cells within the peritoneal cavity also maintain a stem-like phenotype following adoptive cell transfer. Essentially, we observe them as being C39, 69 negative, and also TCF1 positive. And so they maintain this phenotype for up to 3 weeks post-infusion, whereas the 2.7 effector-like T cells really have this much more effector and terminally differentiated phenotype in vivo. Getting back to the role of the endogenous immune system in the response, we ran studies both using lymphodepletion and the use of rag knockout mice that lack mature T cells and B cells, and observed essentially the same thing, that if we look at a host that lacks host T cells, we essentially largely abrogate the effectiveness of FRB T cells. So we know that host immunity plays an important role and is really required to prolong anti-tumor immunity. And so this cartoon sums up really the findings from a study that was published last month in the Journal for Immunotherapy of Cancer, where if we take FRB2.7 T cells, which really have this very robust effector phenotype, the trade-off is really they have very poor persistence following infusion, and therefore we have very short-term BITE-driven activity against the tumor. On the other hand, the FRB2.15 T cells maintain a stem-like phenotype and have enhanced persistence, but within extratumoral spaces outside of the solid tumor. And from here, they're able to secrete BITEs and really redirect predominantly host T cells for prolonged BITE-driven attack on the tumor. In the second part of my talk, I wanted to talk about some additional opportunities for FRB T cells that we're currently exploring. So the first is really related to the observation that FRB T cell therapy can broaden the anti-tumor T cell response. So the I9MP1 folate receptor alpha model also expresses OVA or ovalbumin as an additional targetable antigen. And what we noticed was that when we treated animals with FRB T cells, we saw increased expansion of OVA-specific CD8 TILs following FRB T cell therapy. I'm just showing you a tetramer staining here for the immunodominant epitope of OVA. We suspect, given the non-specific nature of BiTE redirection, that this is not driven by preferential expansion of tumor-specific T cells, but is instead driven by epitope spreading. That is, destruction of the tumor, release of tumor antigen, and then direct presentation of that antigen by the host to these T cells. And so we think this is important for a couple of reasons, and there are additional ways we can leverage endogenous immunity beyond just the response to the secreted BiTEs. So first, this is an opportunity to overcome tumor antigen heterogeneity. As I already showed, not all ovarian cancer cells are going to express folate receptor alpha, even in the high expressing patients. And so if we can broaden the T cell response, this lowers the potential for outgrowth of antigen loss variants due to tumor antigen heterogeneity. Additionally, because these host T cells and these endogenous T cells have a better chance of persisting long-term, even after the T cells are cleared that we adoptively transferred, we see this as a clear opportunity to produce durable and lasting tumor attack, even once the adoptively transferred T cells have begun to contract. And so presently, we're working to understand whether FRBH T cells, that is the human version of our therapy, can also drive expansion of tumor-specific T cells in ovarian cancer patient specimens. The second way we're thinking about using BiTE-arm T cells is rationally combining with other forms of immunotherapy. And the data I'm going to show here is related to checkpoint blockade. So in ovarian cancer, despite promise and other indications, the response rate to single agent checkpoint blockade is very minimal, typically on the order of less than 15% objective response rates are reported. And from the data we've seen and from our own data, we suspect this is really related to a lack of tumor-specific T cells present within the tumor microenvironment, which we suspect we can overcome through BiTE-mediated redirection. Furthermore, we've observed that CD8 TILs upregulate PD-1 in response to FRB T cell delivery. And this is true of both the engineered T cells shown on the left, but also the endogenous or bystander T cells shown on the right, providing clear rationale for combining FRB T cell therapy with checkpoint blockade. And to first establish this as proof of concept as a combination, we developed an in vitro model where we cultured human T cells with folate receptor alpha expressing PD-L1 positive SCOP6 tumor cells. And we observed that the addition of anti-PD-1, here we're using a pembrolizumab biosimilar, can improve both target cell lysis at multiple effector to target ratios, and also improve the function of PD-1 positive FRBH T cells. And so based on this data, we then went on to test this combination against ovarian cancer patient specimens that have been collected from patients here at Roswell Park. And what we observed with this combination was really three different types of response. We saw in some cases that patients are non-responders. And here, for simplicity, I'm just showing the ovarian cancer patient specimen plus or minus IgG and PD-1, and then the FRBH T cells plus IgG or anti-PD-1, not the control cells. So in some cases, we see non-responding patients. In other cases, we see patients that respond to the FRBH T cells, but really show no further benefit from the addition of anti-PD-1. And then in other patients, we see a response to the FRBH T cells that's further magnified through the addition of the pembrolizumab biosimilar. When we began to look for baseline correlates of response that would help us to understand sort of these single agent responders versus combo responders, we again saw that folate receptor alpha expression really correlated with response. So this non-responder is a folate receptor alpha low. All the responders really fall into the folate receptor alpha intermediate or high category. But when we looked at PD-L1 expression at baseline, either in the tumor cells that were present or the immune cells that were present within the tumor microenvironment, there was really no clear correlation between single agent responders and these combo responders, which told us it was a bit more complex than just looking at antigen expression and PD-L1 levels to determine response. In parallel, we've also been working in preclinical models combining FRB T cells with anti-PD-1. And very similar to what we observed in these patient specimens, we see that some animals really benefit from the addition of PD-1 and the response to FRB T cells is enhanced, whereas other animals show no additional benefit to adding anti-PD-1. And when we look at these non-responders, the anti-PD-1 non-responders, we see that they have increased accumulation of multiple myeloid populations within the tumor microenvironment, including neutrophils and monocytes shown here on the right. In comparison, the PD-1 responders have increased accumulation of M1-like macrophages. And so we think that the myeloid compartment likely plays an important role in determining the single-agent responder to the FRB T cells, and then those that can potentially benefit further from the addition of anti-PD-1. And so presently, we're using these two models as complementary approaches to identify strategies of improving the combination efficacy, including identifying which patients are most likely to benefit from the addition of pembrolizumab to the FRBH T cell therapy. The last piece of data I want to discuss is really related to overcoming barriers to FRB T cell therapy. And in this case, I want to touch on the role of the ECM, or extracellular matrix, and its interaction on limiting with FRB T cells on limiting their efficacy. And so the data I'm showing here is transcriptomic data from the effector-like FRB27 and the stem-like FRB215 cells 5 days post-ACT. And while the stem-like cells have signatures of T cell replication and T cell function, the FRB27 effector-like cells had strong signatures of ECM interaction. And so we hypothesized that this interaction with the ECM as T cells are differentiating this effector-like phenotype may limit their ability to persist and also to function within the tumor microenvironment. To test this, we co-cultured FRB27 effector-like T cells with FR alpha positive target cells in the presence of increasing amounts of plate-bound collagen. And what we observed was, as exposure to collagen increased, the ability of these cells to effectively target cancer cells decreased. And to overcome this barrier, we've now been working on a multi-armed T cell strategy where FRB T cells are additionally outfitted with a FAP-targeted CAR. And so this FAP-targeted CAR is designed to target fibroblast activation protein, or FAP-expressing CAFs, or cancer-associated fibroblasts, which are really a major source of ECM deposition in tumors. And so what we've observed is that the multi-arming of these T cells does not inhibit the ability of the cells to secrete BiTEs and drive FR alpha targeting. But now we have the additional benefit shown on the right here of also targeting FAP using these multi-armed T cells. So just to summarize what I've shown today, FRB T cells produce robust therapeutic activity against FR alpha positive cancer and redirect endogenous T cells through T cell engager secretion. FRB T cells can persist and direct anti-tumor immunity from tumor-adjacent or extratumoral spaces. In ovarian cancer, this would be the peritoneal cavity. And this suggests a unique mechanism of action for BiTE T cells that doesn't have an absolute requirement for direct infiltration into the tumor to drive therapeutic responses. We've observed that treatment with FRB T cells leads to expansion of tumor-specific endogenous T cells. And this has important implications for overcoming not only tumor antigen heterogeneity and outgrowth of antigen loss variants, but also in generating durable and curative responses potentially for patients. FRB T cells can rationally combine with multiple forms of immunotherapy, for example, checkpoint blockade. And this is particularly true when low intrinsic tumor immunoreactivity may be limiting therapeutic response. And so we're working to understand how patient selection may be essentially overlaid with the best way to rationally combine FRB T cells with other immunotherapies. And lastly, multi-armed BiTE T cells can be used as a strategy to overcome additional therapeutic hurdles in solid tumors. For example, what I showed you today is targeting the tumor stroma to really unlock the full potential of FRB T cells following infusion. With that, I'd like to move to my acknowledgements. First, acknowledging all the members of my lab, both former and current, especially Jesse Cello, who I think was involved in pretty much every study I showed you today. Our collaborators at Roswell Park, University of Chicago, and McMaster University. All of the shared resources, departments, and programs at Roswell Park who support our research, as well as the funding support for these projects. Thank you very much for your time and attention.
Thank you so much, Robert McGray. I just want to mention quickly the Q&A session is going to be at the end of the second talk, and please direct your questions to the QA box. So the second speaker today is Dr. Mark Yarmarkovich, Assistant Professor in the Department of Pathology at NYU School of Medicine. Dr. Yarmarkovich obtained his PhD in cell and molecular biology at UPenn, where he developed a tumor antigen discovery pipeline using genomic and proteomic data to discover peptide MHC ligands. Then he went to do a postdoc at the Children's Hospital of Philadelphia, where he continued the development of CAR-T and TCR-based immunotherapies. He's the scientific founder of two companies, one focused on immunotherapy antigen discovery and another on clinical immunotherapies for pediatric cancers. And his most recent work has been focused on developing peptide-centric CARs for pediatric cancers with high unmet needs, which I think is the topic that we're going to hear today. Welcome, Dr. Yarmarkovich.
Thank you very much for that introduction, Dr. Hernandez-Lopez. And, this work that I'll be showing is a project that we started at the very early days of the Cancer Moonshot project. So it's really a pleasure to be able to present this as it's come to fruition many years later. So as was mentioned, I'll be talking about our work in targeting intracellular proteins using what we're calling peptide-centric CARs. So I think this audience is well familiar with CAR T cells, but in case you're not familiar with the story of Emily Whitehead, I highly encourage you to read about it. And, I think this slide always powerfully demonstrates the potential of CAR T cells. And so this is Emily Whitehead, who was treated with a single dose of anti-CD19 CAR T cells 10 years after her therapy at my previous institute at CHOP 10 years after her treatment, and this is now being called a cure. So despite these promises, when we look at the landscape of total cancer patients, the successes of CAR T cells have really been limited primarily to the liquid tumors, aside from several case studies. And as was mentioned in the previous talk, patients who benefit from checkpoint inhibitors have mainly been patients with highly mutated tumors. And roughly speaking, that leaves patients with tumors with low to medium mutational burden with few to no effective immunotherapy options. And one of the reasons for this has been attributed to the lack of T cell function in a solid tumor. And to overcome this, the field has been taking several different strategies, armoring T cells with cytokines, with chemokines, using them in combination with small molecules and antibodies, reprogramming T cells genetically or epigenetically to drive specific T cell fates, and then using combinations of metabolites and nutrients, and as well as reconfiguring the CAR receptor itself, modifying the hinge membrane, the transmembrane domain, the co-stimulatory domains, as well as the strategy that you heard in the previous talk. But all of this is really a non-starter without the knowledge of a tumor-specific antigen and the ability to engineer an antigen-specific receptor, which is really the prerequisite step in initiating a T cell response. And so this is the interface where the work that I'll be showing you is focused on, is discovering new tumor antigens and engineering what we call peptide-centric CAR receptors. So this is a landscape showing the majority of targets that are currently being pursued in the field. Most of these are plasma membrane proteins. And in terms of targeting these with high tumor specificity, these membrane proteins are not optimal targets for several reasons. One is that the membrane proteins really comprise only a fraction of the proteome. Of those, very few are actually essential to driving the cancer. And because of that, for example, patients treated with CD19, they can downregulate CD19 or splice out the epitope targeted by CAR T cells to evade the immune response. And as we know, most of the critical drivers of cancer are actually located within the cell, in the cytoplasm or in the nucleus, and mostly these have been inaccessible to CAR T cells to date. However, we know that proteins from the inside the cell can be presented on the surface of the cell through the antigen processing pathway on MHC. So for those of you who aren't familiar with this pathway, briefly, proteins are degraded in the proteasome, peptides are transported into the ER, where some of them are loaded onto MHC molecules that are transported to the cell surface, where they're surveyed by native T cells. And when we look at the interaction between the peptides presented and the T cell receptor, the peptide MHC is recognized as a complex by the TCR alpha and beta chains, in which the germline encoded CDR1 and 2 loops canonically dock on the MHC to position the hypervariable CDR3 loop to recognize the peptide. And intracellular targets have primarily been targeted using immunotherapies through neoantigen-directed therapies, such as checkpoint inhibitors. So this has really been bottlenecked by the number of mutations that are necessary to elicit a durable anti-cancer response. And that's really limited by the number of peptides that are capable of binding to MHC molecules, which is, we estimate, is at around 5%. That's then further limited by those that are immunogenic to native T cells, and that's been estimated to be at about 1.6% by Steve Rosenberg, who's probably looked at the largest number of these immunogenic neoantigens as they respond to TILs. And then really to achieve a complete response, those antigens really need to be derived from clonal drivers of the cancer. And then again, for TCR therapies, that's further limited by the HLA restriction. So specific HLAs will present specific peptides, and that peptide MHC complex is recognized by the T cell receptor. So not only are we limited by the pool of clonal driver neoantigens, but then that's further limited by the subset of those patients that share a particular HLA allele. Now, this work I started in John Maris's lab, which focuses on a deadly pediatric cancer called neuroblastoma. And in terms of targeting peptides on MHC, it really has two major challenges. One is that it's very low in mutational burden alongside most of the other pediatric cancers. And two is that it's actually the lowest HLA expressing tumor. And so while it had these two challenges in terms of targeting peptide MHCs, we thought that by addressing these challenges, we could apply those principles to other cancer types. So neuroblastomas, rather than being driven by point mutations, have recently been shown to be driven by the so-called core regulatory circuit. This is a set of transcription factors that bind one another's promoter sites. And this is a circuit that's on during early development in the neural crest lineage, and then is silenced upon differentiation of mature cells. And the hypothesis that I started with was, since these are non-mutated proteins, can we find these presented on MHC in tumors? And then if so, can we target these as non-immunogenic self-peptides using immunotherapies? So this is just to show a snapshot of the workflow that I'll go through in this talk, and I'll try to cover each of these topics briefly, and this has all been published. And to give away the punchline, this work has now resulted in a clinical trial that we're planning to open next year targeting FOX2B presented on MHC. And that's been really driven by the preclinical results that we've achieved and being able to eradicate these tumors in PDX models. And so I'll start with how we started with tumor tissue, discovered these targets, validated them, and then developed these peptide-centric CAR receptors. So we started with a workflow called Immunopeptidomics. Here we start with tumor and normal tissue. We purify the MHC off of the surface of these tumors, and then we elute class 1 and class 2 peptides, and then do tandem mass spectrometry to identify the specific peptides presented on these tumors, as well as the landscape of peptides that these tumors globally present. We then integrate that data set with NGS data, so genomic and transcriptomic data sets, both from tumors as well as normal tissues. And then we developed a computational pipeline to prioritize these peptides for those that are tumor specific. And that let us go from about 9,000 peptides that we found in neuroblastoma to about 50 that we think are highly tumor specific. And to show you an example of one of these, this is the lead target FOX2B that I mentioned is going into the clinic. In the first column here, we're showing expression in about 150 neuroblastoma tumors. And then the subsequent columns show expression in normal tissue across about 1,600 normal tissues grouped here by different organ sites. And so this is exactly the type of profile that we look for, a very high expression in tumor, and then low expression across different normal tissues. And then to contrast that with a well-known immunotherapy target, HER2, you can see that there's quite a bit of normal tissue expression in this molecule. And this is really, although been successfully targeted using monoclonal antibodies, has not had that same success using CAR T cells and has actually seen some toxicity in the clinic. And that's likely due to this normal tissue expression you see here. And so while we think this target has a better therapeutic window, there are some major challenges with targeting these peptides on MHC that I'll discuss. So going back to FOX2B, again, from that core regulatory circuit, FOX2B is actually a member of that circuit and is necessary in driving these neuroblastoma tumors. And consistent with its biology as a developmental transcription factor, you can see that FOX2B is highly expressed during early development and then completely silenced before birth across various tissues. And we know from DepMap that FOX2B is actually the third ranking dependency gene in neuroblastoma. So these tumors really don't tolerate the depletion of FOX2B. And that really, we think, cuts out one potential mechanism of immune evasion to this target. Now, the challenge of this targeting this peptide on MHC is nicely illustrated by the crystal structure that we solved. So this is showing the FOX2B 9-mer peptide. This is showing that 9-mer peptide in the context of the HLA that presented. So really the tumor-specific portion of this complex is the 9-mer, and this MHC molecule is shared across all tissues in these patients. So our challenge here is really to develop a receptor that can specifically recognize this peptide in the context of a much larger MHC molecule. And the second degree of complexity here is that even if we're able to develop a receptor that can specifically recognize the peptide, if the motif that's recognized by that receptor is also shared across normal tissues, that can also result in cross-reactivity. And unfortunately, that's been demonstrated in the clinic with a T-cell receptor targeting a molecule called MAGE-A3, which is a tumor-specific antigen in melanoma. However, what was done in this trial was that this TCR was affinity enhanced to increase its potency, but in doing so, they had circumvented negative selection in the thymus and introduced novel cross-reactivities. And unfortunately, this TCR cross-reacted with a peptide that was presented from the titin protein presented in heart tissue, that resulted in the death of the first 2 patients treated with this TCR. So we really wanted to get ahead of this type of cross-reactivity, and especially because we were engineering completely synthetic receptors. So we developed this algorithm to predict these cross-reactivities and screen for that at the first step of CAR development. So without getting into the details, I'll show you the validation we did on this known cross-reactivity between MAGE-A3 and the titin protein, and we were actually able to predict that cross-reactivity as the 4th ranking cross-reactivity out of over 1 million potential peptides presented on that HLA. And we were also able to point to expression in heart tissue as a potential liability. And so we thought this tool was useful, and we used it in our first steps of screening CAR receptors. So shown here are a panel of predicted cross-reactive peptides to FOX2B, shown in the different columns, and we screen those against a panel of different SCFV CAR receptors. And what we found is that most of the CAR receptors cross-reacted to at least one of these predicted peptides, which gave us more confidence that the algorithm was pointing us in the right direction, but it also allowed us to quickly hone in on these receptors that had very nice clean binding profiles just to FOX2B with minimal cross-reactivity to these cross-reactive peptides. And this is what they look like by flow, with FOX2B on the x-axis and the cross-reactive peptides shown on the y-axis. And while these predictions were encouraging, the real test is when we take these CARs and co-culture them with HLA-matched tissue that present the full milieu of peptides on MHC, which can include thousands or even tens of thousands of unique peptides. And when we do this, we see that these CARs have no cross-reactivity against these HLA-matched tissues. And then the real test is, of course, going back to the point that these are low HLA expressing tumors, and the paradigm in the field has been that CAR T cells, unlike TCRs, really require a much higher density of antigen, which can be an order of magnitude or 2 higher of a threshold as compared to T cell receptors. So we were unsure whether or not these low HLA expressing tumors would present sufficient antigen to trigger a CAR response. And we were very pleased to see that when we took these neuroblastoma cells, co-cultured them with these CARs, we were able to achieve complete tumor killing. And then to really show that that effect is mediated through FOX2B, we took these FOX2B negative HLA matched lines, transduced them with FOX2B, and showed that we could rescue the killing that way. And then this is showing a couple of killing videos with the 2 target cell lines on the left, and then the 2 HLA-matched tissues on the right with the cleaved caspase marker labeled in red. So we next took these CAR T cells into mice using patient-derived xenografts, including this very highly aggressive COG564 line. And when we treated with these CARs, we were very pleased to see complete eradication of even these very aggressive tumors. And this was actually very surprising to me, particularly for this COG564 line, because if you look at the HLA expression, in this case, this is looking at day 10 of the group treated with untransduced T cells, you can see that these tumors are almost completely negative for HLA. However, when you look at the group treated with the peptide-centric CARs, you can see a very potent upregulation of HLA expression. And so what we think is happening is that the CARs will enter the tumor, they'll see a low density of antigen, and before they start killing, they'll start secreting interferon gamma, which will upregulate MHC and basically initiate a feed-forward loop of antigen density. And that likely explains this lag phase before these CARs are able to overtake this tumor in the COG564 line. Now, to switch gears to a different story, and I'll try to go through this quickly. This is a computational project I did trying to model early immunogenicity using the TCGA data. And one of the early analyses I did was to map the mutational frequency of different point mutations against the predicted population scale presentation of that mutation as a neoantigen in the population. So plotted here on the x-axis are the predicted frequencies of a particular neoantigen being presented in the population. So you can see that some of these neoantigens are predicted to be presented by the vast majority of the population, and these are very lowly represented as mutations in the TCGA dataset. However, what we saw is that there's a very striking enrichment of these mutations that are not predicted to be presented highly on MHC. These are very highly enriched in mutational frequency. Now, when we look at this at a protein level, this is showing the same analysis, this time across the span of individual proteins, with the bars representing the proportion of the patient population predicted to present a particular region of a protein. And overlaid on that are the mutational frequencies along the span of these across the protein. And so what we saw here is that there's a very striking enrichment in these hotspot mutations in these so-called immunogenic valleys that are lowly presented on MHC or not presented at all. And what was quite striking from this is that there was such a disparity in predicted MHC presentation across the population, with some regions of the protein being predicted to not be presented at all in any patients, whereas other regions where mutations were less common were predicted to be presented very highly across the population. And so this got me thinking about this hypothesis, is that if we're finding FOX to be presented on a single HLA allele, being the last step in antigen presentation where the peptide meets the MHC and binds to it, if that same peptide could potentially bind to other HLAs, could it then be presented? And if so, could we target that same peptide in the context of other HLAs using the same peptide-centric CAR receptor? And so this is the same analysis now looking at FOX at the span of FOX2b. This is the 9-mer peptide that we empirically identified by mass spec originally on HLA-A24. But here we saw then in addition to A24, this same peptide was predicted to be presented by up to an additional 8 HLA types. And so we modeled this and tested it. And without having much time left, I'll just say that we also found that this peptide-centric CAR is able to bind this same FOX2b peptide in the context of multiple HLAs. And not only can it bind, but we also showed that it's functional both in vitro and in vivo, where it's able to completely deplete these tumors expressing these other HLA types. We've since solved the crystal structure of the peptide MHC in complex with the scFv that binds it. And this very elegantly explains the basis of this cross HLA docking on MHC and binding to that to the FOX2B peptide in a peptide-centric manner. And so this has been posted now on bioRxiv if you're interested in the story. And so to summarize this approach, we think the advantages of using these peptide-centric CARs are that we can go now beyond the membrane proteins into the intracellular space, being able to target not only cytoplasmic proteins, but also nuclear proteins, including transcription factors that have previously been considered undruggable. And in addition, unlike the TCR, which has evolved to recognize the peptide MHC as a complex, here we're really engineering these receptors to bind to just the peptide and not react against the HLA itself. And so by virtue of that, we're actually able to target that same peptide across multiple HLA types. And we're now really digging into the extent of this effect and looking at new engineering strategies to expand on this. And so to summarize, we've been able to discover new targets presented on MHC, including those derived from previously undruggable master regulators, including FOX2B. We show that peptide-centric CARs allow us to target intracellular tumor antigens, and that these peptide-centric CARs can target non-mutated self-proteins. We show that they can potently and selectively kill neuroblastoma tumors and break conventional HLA restriction. And as I mentioned, because of that, we're now entering a clinical trial targeting FOX2B across multiple HLA types and are now applying this pipeline to other cancer types. And so just want to thank a great group of people that I work with, both in the Maris lab and our collaborators across Penn and CHOP, Immunotherapies, with whom we worked on developing these receptors, University of Tübingen, which helped with initial immunopeptidomics studies, and the great group that we've worked with across the Stand Up to Cancer, and of course, the Moonshot program, which really made this work possible. So I'll put my email up in case anybody wants to ask questions, and I'll make a plug that we're also recruiting at all different levels if anyone's interested in this type of work. So I will stop there. Thank you.
Thank you so much, Dr. Yarmarkovich. Now we're going to start the Q&A session. And please, I'll remind you, just input your questions in the corresponding box. So the first question is for Dr. McGray. They are asking whether the bites were injected intratumoral or IV, and whether the endogenous immune system is engaged. Did you observe any abscopal effect?
So the bite engineered T cell. So in this study I presented today, we're not infusing soluble bites. It's only T cell delivered bites, but they were given local regionally, intraperitoneally. So we actually think in ovarian cancer there's a unique opportunity to deliver local regionally by IP infusion. We've done this with solid tumors as well in other sites in terms of local delivery. We're beginning to explore also systemic delivery, but we think that because one of the goals is to not lymphodeplete, we know that expansion is going to be limited of the T cells. And so we think giving them locally gives them the best chance to be active versus systemic delivery, but we haven't really tested that as far as to make really strong conclusions around that. As far as abscopal effects, we haven't tested that in an appropriate model, but I think it's a great question. So certainly we'll look into it.
Another question for you that might be related. So I'm gonna put them together so that what would be the advantages of the T cell expression of bites over either B cells expressing bites or CAR Ts, I guess?
Right. So I mean, one of the real advantages of delivery from a T cell is the T cell that secretes the bite isn't only secreting it, it also can respond to it. And so the T cells themselves also respond to the secreted bites. In fact, when we look at in vitro studies and we look at proximity, the T cells that secrete bites do react or engage more strongly than the bystander T cells. So we know that there is a proximity advantage to the T cell. After infusion, T cells are also really among cells that are going to expand. T cells expand exponentially compared to other types of cells, and so we would suspect that T cells really are going to expand and drive a much more potent effect than in engineering a different type of cell. And so we think that for those reasons, the T cell is probably an ideal platform. The part around a CAR, I mean, certainly we think that, multi-arming is certainly an option. I mean, we're currently exploring that as a strategy in terms of whether delivered from a CAR or the T cell itself. I think both are options, but we think the T cell really is a very advantageous vehicle for delivery.
Great, thanks. There is a couple of questions here for Dr. Yarmarkovich, and then I'll go back to one that I think it's for both of you. One for where it's about the initial group of binders for the selected peptide. Like, how did you start, I guess, generating the scFv against FOX2b, and the other one, or if you want to go first about that, and I'll ask you this.
Yeah, so I didn't really have time to get into that. So those binders were pulled out of a phage library. So the library had about 10 to the 10th scFvs, and that part we worked with a couple of collaborators on that have various libraries, synthetic and naive libraries. So the binder that I showed, 10LH, was from a library from Immunotherapies, which has really developed a nice screening platform for peptide MHC binders, where they're able to identify these very rare binders that have the right selective properties.
Great. Another question, it's related to the cancer evasion mechanisms, no, and the downregulation or loss of MHC expression. If that happens, I guess the PC CARs will be blind to it. So what would be the mechanisms to avoid that?
Yeah, I mean, that's been the major concern going into this work. And, of course, that is the main mechanism of immune evasion we're looking at. Luckily, we haven't seen that yet, at least in our PDX models. It's something we'll definitely be keeping an eye on. And as I mentioned, one of the other main concerns was the low MHC expression. And we were looking at strategies to preemptively upregulate MHC, but, so far it seems to not be necessary, as the T cells are actually doing that part for us by secreting cytokines in the tumor microenvironment. And then, the other thing is that these neuroblastomas are very lowly mutated tumors, and so the hope is that because they're because of this low mutational burden, they'll be less likely to evolve mutations in the MHC pathway. And while that is the most common mechanism of immune evasion, the good news is that it usually doesn't happen.
Thank you. And now there is a question for both of you. So probably I'll go back first with Dr. McGray, and then with you, Dr. Yarmarkovich. So what is in terms of the differences in terms of signaling in the T cells when they are activated by a BiTE or a PC CAR? How would that compare to the regular, TCR signaling of T cells?
Sure. So, I mean, I think that there's some data, certainly the data is not from us, it's published previously, but that, the immunological synapse formed by engagers is a bit more similar to the TCR, and the downstream signaling is a bit more physiological. But we suspect that given that we are activating with a single chain antibody, we know we're not fully recapitulating all of the biology of the TCR. So we're certainly thinking about those kind of things, but we do see that, certainly the T cells can, go through multiple rounds of stimulation. They can kill in sort of chronic stress assays and sort of, repeated chronic antigen stimulation. So we suspect that it does recapitulate some of the aspects of TCR, but we're looking into sort of what defects may exist, including concepts of co-stimulation and how that might play a role as well.
Yep, and I completely agree with Dr. McGray's comments, and I think there's advantages and disadvantages to each approach. The advantage of the BiTE is that it signals through the native T cell receptor, which is less limited than the single co-stimulatory domain of these CARs that we're using. I think the advantage of the CAR is that you can get a memory T cell response, whereas BiTEs, you have to dose repeatedly. So I think each system has its pros and cons and different applications.
Thank you. Here's another question also for both of you. So maybe you can start, Mark, and then Robert. What are the implications of these designs or approaches that you are proposing in relates to their potential synergy with neoantigen vaccines? Can you comment on that?
Did you want to flip the order this time or same order?
Yes, yes.
For our approach, I think it's really directed at the tumors that don't respond well to therapies like checkpoint inhibitors or vaccines that rely on a high mutational burden. I think the key advantage is that we're able to use this approach in non-mutated cancer drivers. They could also be applied to neoantigens derived from mutations, but as you point out, those can be targeted through other approaches like cancer vaccines or checkpoint inhibitors.
So one of the things that we are, it's some new data, but we're particularly intrigued about is the idea that we actually see this broadening response when we use engagers delivered from T cells. And so, that may be a clear opportunity to combine this type of therapy with neoantigen targeting. So, I mean, we've only generated data in mouse models thus far, but typically robust epitope spreading has not been commonly described for CARs. And so we think that there may be some uniqueness to the mechanism that we're observing that we could potentially enhance or improve on or boost given, using vaccine approaches. And so certainly something we're thinking about, but certainly in the context of patients, it would be important to see it there as well, and then sort of understand that as an approach clinically.
There is a couple of other questions. It's not clear who they are related to, but it says CAR T cells are often short-lived in vivo. Is there any study to check on anti-idiotype responses that might eliminate the engineered CAR T cells?
Yeah, I mean, in that first slide I showed with, looking at CAR T cells 10 years later, that there was actually an analysis done for those first group of patients, and they do find memory T cells even 10 years later that are still functional. So, there is an antibody response that's been shown against the CAR receptor, but it seems to, not preclude an CAR activity even many years later. So you do find memory cells persisting for a long time.
Great, thanks. Here's another question, if I may ask, related to both of you. Like, in terms of the comparison of TCRs and BiTEs and now peptide-centric CARs, do you find any correlation in terms of the affinity that you have to tune either the BiTEs or these scFvs, to have like the corresponding or best response and in comparison to TCRs and CARs?
I'll let you take this one. You're the expert in affinity here, I think.
It's a complicated question. I think in general, the trend is that affinity enhances activity, but we do find a lot of outliers that, where we find very high affinity receptors that don't activate the CAR, or low affinity receptors that actually work better than some of our higher affinity receptors. So I think in general that principle holds, but I think there's a lot we don't understand about the mode of binding and the geometry that really can dictate binding that somehow breaks these rules. So I think there's a lot to be learned still about how that affinity and mode of binding correlates to activity.
Just to add one point, I agree completely with that, with your response. The one thing I want to add is just when we've been talking with our colleagues about this as well, that often even when we make modifications to a single molecule that we've been developing, whether it's changing linker lengths or changing just different aspects of the design, that really we have to really empirically be testing these things. It's very challenging to actually predict that a high affinity interaction is going to lead to better T cell activity or less T cell activity for a weaker affinity. And so I think that the challenge that we're thinking about, and I think everyone's thinking about, is how do you accurately predict those things without doing the appropriate wet lab studies.
So with that, I think it's time to thank our speakers, Dr. McGray and Dr. Yarmarkovich. Thank you. Thank you to our moderator, Dr. Hernandez-Lopez. I appreciate your time, and those are excellent presentations. And our next Cancer Moonshot seminar is August 24th, spatial data analysis and AI models for cancer research. Thank you, everyone. That was excellent.
Thank you.